types of technologies may in fact be weaponizable. And what I mean by weaponizable is that they can be used in the classical definition of a weapon, which is a means of contending against another so as to determine, access, or decrease their will, morale, or intention to fight or wage harm. Now that said, that doesn't necessarily mean that all the things that I'm going to talk to you about tonight are in the general employ of governments and or the government, because then I'd have to kill you before we leave. And number two, I'm going to also tell you that in certain instances, some of the work represents what we call trajectory science, which means that it's on the trajectory of being almost out of the oven, but it's at a pre-translational stage in terms of the way we're actually going to use this operationally. That doesn't mean it's not going to happen. That doesn't mean it's science fiction. It means that we're not quite there yet. And the third concept goes along with the first AAT concept. Obviously, if we can assess something, which is the idea of what most of these technologies will do, if we can then access it with regard to being able to utilize our technologies to then gain this particular site in the brain or elsewhere in the nervous system, then we can target it for something. And I'm going to tell you what these things are really used to be targeted for. The idea is if we can predict, then we may be able to intervene. And if we intervene, we do so towards prevention. Which is why the specter is of this fictional construct known as Minority Report. I'm not a real big Tom Cruise fan of the film Minority Report because I think there were some things in there that were just like, oh, way out of... Now it's going to go on YouTube that I'm not a big Tom Cruise fan. I like Tom Cruise. I think he's a great guy and he's a good actor. But there were some things in this particular movie where, you know, it goes, oh gosh, that's terrible, horrible, miserable. Yeah, right, that's called science fiction. One of the things that might happen is as you leave here tonight, you may be scratching your head and go, how do we stay off of that slippery slope? And that's okay if we do that. So, let's jump in real quickly. Once again, if I say anything that is jargon-esque and you don't know what I'm talking about, I apologize and simply stop me, just call out, raise a hand, jump up and down, hit me with food, that's fine. And the other thing is, seriously, if you don't understand my New York accent, well, I can't help that, but I'll do the best I can. So, we'll start off from the premise of this thing called neuroscience. I've spent the last 32 years of my life doing this. For those of you who had the opportunity to read a brief introduction of what it is that I do, I spend some of my time in a very strong laboratory circumstance doing basic and clinical translational neuroscience, mostly molecular neuroscience, that has a very strong bend towards what's called computational neuroscience. My actual bread and butter is that I'm a pain researcher, which is creepy enough as it is. And then the rest of my time I spend at a policy institute here in the district called the Center for Neurotechnology Studies at the Potomac Institute for Policy Studies, where we're looking at cutting-edge science and technology based upon our former pedigree. The Center for Neurotechnology Studies and the Potomac Institute get their pedigree from the fact that Potomac was at one point the Office of Technology Assessment that was decongressionalized in 1994. So the idea is really taking a deep dive into science and technology towards understanding the science and tech, to then being able to understand the ethical, legal, and social issues that arise from their use, misuse, inappropriate use, and then that's what informs and perhaps may formulate policy. Now, I won't get into the meat and potatoes of the policy, but I will get into some of the meat and potatoes of the ethics, because that's really the idea where the rubber hits the road. So if we talk about this science, neuroscience, first thing you need to know is it's a relatively new science. In fact, the term neuroscience wasn't even around when I went to school. The program that I went into was not a neuroscience program. It was a program in physiological psychology, because the first neuroscience programs didn't pop up until the middle 1970s, which should give you an idea about how old I am. And so what happened is, in a short amount of time, do the math, from about 1974, first real discrete titular program in neuroscience, to the present, now you see this booming field that brings together a whole host of different disciplines under the singular rubric of being able to study the way the brain and nervous system works in general, and by extension and focus, particularly the human brain and human nervous system works, to modulate a whole host of things, sensations, perceptions, and then at this level, cognitions, emotions, intentions, and behaviors. Obviously, contemporary neuroscience has made tremendous leaps, as I've tried to illustrate for you here by this panel of illustrations that suggest the idea of 17th and 18th century depictions of the brain taking a leap to the contemporary period of a static image, which is computationally enhanced, of the human brain, to the idea of then utilizing these salient characteristics of structure and increasingly of function to then be able to access the brain bidirectionally. And what I mean by this is not only to record from it, but also to then utilize similar technologies in reciprocity to stimulate or inhibit particular brain regions to then control the way certain brain regions and networks may function or not function, and ultimately in that way be able to direct, or in some way, in quotes, and this is a nasty word, control those functions on the level that may range in some cases from the synaptic, the space connecting gap that exists between nerve cells, all the way to the psychosocial. So one of the things that I want to ground you to is how this field has gotten to this point, because this also talks about the potential for the way that we use neuroscience and its technologies. There's a particular lesson learning that goes on with much of science and the technologies that are used to get the science and then evoke the science as a task. And these are called, sometimes referred to as Gigerenzer heuristics. And what it really refers to is, first, we have a set of tools that are at our disposal. And we use those tools to study the natural world, in this case, the natural phenomenon of nervous systems in the brain. That allows us a particular level of theoretical acumen. We develop theories, how things work, what they're all about, what they mean. Once we get those theories, we then develop more sophisticated tools that we then apply to test those theories, supporting some, subordinating others, and yet negating others. And this then becomes this reciprocity of tools to theory and theory to tools. And these are the heuristics that built neuroscience into the science it is today. But more than this, contemporary neuroscience has cracked many of its silos of specificity. We don't see in neuroscience pure pharmacology, pure molecular biology, pure psychology any longer. These are increasingly being de-siloed to open up an opportunity space for collaboration in this new process which is known as integrative scientific convergence. What this allows for is a much larger opportunity space to tackle bigger and bigger problems, utilizing the tools and theories and applications of this science called neuroscience that spans from the submolecular all the way up to the mass cultural. And in fact, this has been the trend and trajectory that neuroscience has assumed increasingly over the past 10 years as a consequence of this snowballing effect that we found. So one of the things that we need to understand is that whenever you see rapid and demonstrable progress like this, two things need to happen. First is we need to sort of sit up and take notice of what we can really do. Second is we need to sit up and take equal notice of what we really can't. And far too often what tends to happen is we run the risk of going in one of either of two directions. Over-exaggerative claims of what this stuff really can do and what it means, and we'll talk about that in some detail, or underestimations of what this stuff may really be able to do. And in either case, we run the risk of committing errors of either omission, commission, and all the ethical legal garbage that then gets tacked on board when we put these errors out into what I would call the social cultural marketplace and try to use these things. So what this then brings us down to is what I would call the mantra for the way I approach this. We have to be prudent with regard to the way we study these things and our uses in practice. And if you get no other take-home message from this talk that I give you tonight, some of these things, as I say, are certainly provocative, many are contentious, some may actually scare you. The idea here is that this mantra of prudence creates a set of marching orders that will dictate the pace, progress, trajectory, slope, and valence of neuroscientific and neurotechnological acumen and progress for the next several years. And at very, very least it should. Oh, why? Well, one of the things we're able to see is that in a very short amount of time, in a very short amount of time, These tools of neuroscience have made ardent strides in capability, where there's been an increase in sophistication because of the convergence of a variety of techniques and iterative improvement in the way these particular tools are able to do the job they do, based once again upon an increased understanding of the theoretical basis of the way brains work, and also an improvement in the actual tools that we have at our disposal, not the least of which increased computational capability, an increased set of variables that we now are understanding about various molecular targets, and better insight to get things down to a much more finely grained level of perception and analysis. One of the areas that's been probably most important is being able to assess what the structure and function of nervous systems and brains are doing. And of course, I think one of the fields that you probably see overwhelmingly, even in your daily lives, is the fact that we've seen a number of different forms of imaging that have gone from very, very static images of the brain to much more dynamic representations of putative or hypothetical functions of the brain that try to elucidate and demonstrate what different regions of the brain may be involved in, the extent of their particular involvement, types of dysfunction that may be reflected in patterns of blood flow or metabolic use of a variety of different chemicals, some injected, some endogenous. And so what we see is that this area of neuroimaging has gained particular sophistication, but it too has limits. There's some real problems that go along with neuroimaging, and for those of you who are aware of it or may use it in your practice, you recognize that there's a lot of stuff it can do, and there's a lot of stuff it can't. And being able to parse the difference between those two speaks volumes when we're dealing with making particular predictions, not about whether or not there's something existing in the head in terms of a structure, but what particular functions may mean to the way we then appreciate them and apprehend them in social contexts that deal with normality, abnormality, rightness, wrongness, goodness, and badness. So it opens up a proverbial Pandora's box in many ways that we'll discuss in some more detail. But this represents sort of the contemporary palette of the major forms of neuroimaging that we have at our disposal to do this kind of stuff. Now, many of these are also usable in the clinical realm, and in fact the translation from clinical realm to non-clinical realm to what we would call social diagnostics is fairly easy these days. Many reasons because of that is that you find a good interface between clinical medicine and we call applied science in those areas such as social neuroscience and its applications in the socio-cultural, even political realm. Well, this is sort of an increasing palette that has gone from relative simplicity, computerized tomography, which is really built upon X-ray principles, a very static image that can be enhanced, for example, through the use of particular types of contrast, to the injection of particular types of radioisotopes that then create much more of a viable signal with regard to what areas of the brain are more metabolically active, particularly by getting a metabolic signal that's derived from glucose and various forms of glucose. We can then increase that a bit, and I'll explain these to you in a little bit more detail, but not to the point where you just fall asleep and you're down in the weeds snoozing away. And then the next more sophisticated level is something we call SPECT, which is single photon emission computerized tomography. It too is a form of X-ray derived tomography, an X-ray type of image, but what it does is it uses a particular type of decay of a molecule that then enhances the signal both with regard to its temporal characteristics and spatial characteristics, gives us a little more fine granularity. We have magnetic resonance imaging that gives us a nice and very beautiful anatomical picture of structure. But structure does very little to tell us what's going on with regard to function. So what we've then done is we pumped up the volume a little bit, and we then have this beast called functional magnetic resonance imaging, which has become so popular that has a number of limits that I'll discuss that have been delimited by the use of what we call co-registered technologies, inclusive of this thing called diffusion tensor imaging, which then measures water molecules and their particular spin in response to a magnetic pulse that help to give us beautiful images of white matter tract connections in the brain. Still some limitations. And if we then couple these types of pure images to electrical activity, at least at the superficial levels of the brain, we get a nice co-registered image that shows structure, function, and physiological characteristics. that bridge some of the gaps between spatial and temporal resolution and non-resolution. So the way we're really seeing this these days is that these things are not necessarily used in isolation, but are used in sort of a pick 'em choose 'em, pretty much the same way you have here on your menu. I don't see everybody just piling up the plate with one thing. We create a complete meal that has an appetizing effect and satisfies what you need and what you want by combining the elements that you're able to get from the proverbial menu of things that are currently available to you, and doing so in such a way as to satisfy the needs. A little bit of ketchup helps, a little bit of chicken helps, a little bit of French fries help. We put them all together and bada bing, bada boom, what do you get? The complete meal of neurotechnology that allows imaging access to the brain and at least some of its functions. But that's not enough. What if you came back to me and you said, but tell me about the recipes. I want to know what they're serving even before I get to the restaurant. I want to know what's going into that stuff and what all these ingredients, when put together, will become. I say to you, perhaps no problem. Because what we can then do is we can take this particular form of neurotechnology that harnesses its power and impetus from the Human Genome Project and a history of molecular biology and genetic techniques, and we can do some nifty stuff here, taking a look at genes of both populations and individuals, as well as the proteins that they express. So if we see these things, and this is a real nice pretty picture, incidentally, the brain you're looking at is mine, and we'll talk about it in a moment. This way I don't have to give anybody else credit. This is my head. I'm not going to tell you what I'm thinking about until later. So what do we find? What has been the level of technological enablement? Well, it's this. It's neuroimaging, neurogenomics, and genetics. Genomics deals with populations, genetics deal with individuals, and the idea that we can put all these things really niftily together, harnessing the power of cyber technology, of computational technology, through the use of ever-increasing data banking that increasingly relies upon cloud computation to pull down tremendous amounts of information in real time. And I'll show you why that's important. Now, what does that allow us to do? Right now, the intent and much of the capacity allows us to do these three things. It allows us to make some nice correlations between structures and functions, not perfect, but pretty good. It allows us to take at variations of what these structure-function relationships look like within populations and individual predispositions. So we see this is what a norm looks like, this is an abnorm, you're fitting somewhere in between these two, sort of like the way we look at an x-ray. I know what a femur looks like, yours has a big crack in the middle, not good. So we see something like this and we go, not good. It's great when you see a stick in somebody's head or the tumor the size of a softball. It's a lot harder to do when we're trying to figure out network patterns in the way things function. And it gets really tricky when we go to this level and we say, what about individual trajectories? What people are gonna do based upon their current level of structure and function and what their genes and proteins tell us they might do. So you can see that what I'm doing is digging a very much deeper hole for neuroscience and neurotechnology to have to try to dig itself out of by means of the explanatory power of what these things do. So let me just sort of give you a little explanation of what these things do, what they can do, what they can't do. First of all, they're really pretty pictures. And you've seen tons of these, I'm sure. You can't swing a wet rope over your head these days without seeing a picture that looks something like this, or the other forms of images that I'm about to show you. And I guarantee this to you. And this is an experiment you can try at home. And since this is on YouTube, you in the YouTube audience can in fact do this in the privacy of your own home. Simply Google brain image and see what comes up. Gazillions of these pretty pictures, and you can go to a magazine in a magazine stand, you can take a look in the newspaper, and I guarantee you, if you spend more than 20 minutes in a magazine stand, not even looking at a science magazine, you'll find one of these. Having to do with this is your brain on drugs, this is your brain on sex, this is your brain when you buy a Ford, this is your brain when you smoke. You've seen these things. Big note to you. Brains don't really look like that when you open them up. All right? Am I right? Okay. See, I'm going to use you as my straight man in the back of the room. I'm going to bounce every neurological question off of you. Been there, done this, seen this, they don't look like this. Moreover, your nerve cells are not color-coded for easy ID. That's a mistake. This is as much art as it is science and skill, because there is a very, very specific computational translation program that allows for It's called a colorimetric translation of individual pixelations of different densities of intensity of signal that are then codified like this. How many of you remember that old TV series, the first one called The Outer Limits? Remember that? Yeah, so it's usually it's a science crowd. You can always tell who the science geeks are. It's like, me, I remember, I've got all the reruns on DVD. Yeah, I love that show. Remember in the beginning the guy would say, there's nothing wrong with your television set. We control the horizontal. Remember that? We control the statistical parametric program that defines what is signal and what is noise. We do it. We do it. We set the threshold as to what we define as a viable statistical signal and what is not. We set the different limits upon which we then figure what is signal and what is noise so as to be able to get a statistically valid representation of what may be going on in these different brain areas. Well, this is positron emission tomography. It's a nice jump up from computerized tomography, and what it does is it really provides a spatial, albeit crude, representation of metabolic activity in the brain. Characteristically, those areas that are more active pop up as red. And this is what's called a standard iconography, a semiotic iconography. We see red as hot. Those are hot brain areas. And what this really represents is the fact that there are, in fact, metabolic changes that are occurring, characteristically in response to the conversion of a primary molecule, which is usually some derivative of glucose. Let me get the next one. Okay. We can increase this a little bit. And we can then ramp up this signal through this thing called SPECT, which is single photon emission computerized tomography, which once again is based upon a tomographic image, which is then mapped to what's known as a standard set of coordinates. These are sometimes referred to as Talairach coordinates, if you hear these noted in the literature. And what we're then able to do is we're able to get differential signals based once again upon metabolic activity as well as cerebral blood flow, because now what we're doing is we're putting in particular types of tracers that identify what brain areas are getting more blood flow at a particular time and what also are the particular metabolic characteristics. Now, we could dig down the rabbit hole a little bit, and this really does represent a somewhat more sophisticated version of what you saw before, which was PET scanning, but then builds upon CT scanning, and is pretty good. But it is weak on two levels. It's temporally a little bit weak. The signal intensity takes a little bit while to come up, and you can still see that these images are pretty crude. These are sort of gross brain areas. That would be like me trying to figure out what the person in the back of the room is having, and the best I can sort of do is I can say, well, at the barbecue pit, I know somebody's having a burger. Duh. Yeah. But getting closer, certainly getting closer. And then you have this. And this is the thing that lights everybody up. And really, when functional magnetic imaging came out, it was a big deal. It was the bomb. Why? Because what it does is it passes a magnetic current in that resonates, changes a particular spin of molecules with response to whether blood is laden in hemoglobin or has a non-oxygenated form of hemoglobin that represents a conversion between blood going to a site and then blood being used at a site. It is represented as showing functional activity in different brain regions as a consequence of what the brain and the person whose brain this is doing. Now, this is one area that has been tremendously exploited. There has been a 400-fold increase over the past 2 to 3 years in numbers of studies that have utilized some form of functional magnetic resonance imaging, either alone or together, and there has been a multi-thousand-fold increase in this type of neuroimaging, again, either alone or together, since neuroimaging was first, in fact, launched as a viable discipline. If we look at the publication slope over the past 10 to 15 years, it's practically vertical. It's practically a straight line going all the way up, with an increasing number of studies that are utilizing this. And in fact, I also serve as an editor for a medical journal. And one of the things that came up in our editors conference was if, in fact, people are doing these types of central nervous system studies, if they're not doing something where they're literally sticking something in the head and they're trying to determine that there's brain regional activity, without these types of pictures, that's just not going to get published. Moreover, the number of journals that are dedicated to neuroimaging has increased 300-fold. Now that's a lot. That's really a lot. And if we take a look at this image, this really does look like a very nice image. Why? Because we see that there's a fair amount of spatial representation and tightness. These are brain areas that are really, really hot. This is a brain where the nose is up here, right, and the back of the head is down here. This is somebody, I'm not going to say who, me, facing this way. And obviously, this is the back of the brain, and this is an area we know, based upon anatomical and physiological studies, to be involved with something visual. So one could look at this and go, well, there's a visual task going on here. There's also an area here, which is in the left temporal junction, which has to do with some form of recognition and identification. And there's also another area here on this other side, which is sort of the non-dominant side in most people, but not me, because I'm left-handed, which is why it's probably lit up like a candle, that has to do with emotionality, what we call prosodic emotion. What I'm actually looking at here is hard to say. It's either a glass of bourbon or my wife. I don't really remember. The response is the same, and they merge depending on more I've had either. But what we find is that there's an increasing amount of inference that says we might be able to take a look at this type of an image and do three things. Number one, relate these back to studies of similar brain images to try to determine what this brain image is actually telling us that this person may be doing. Then relating that back to whether or not this represents some norm or an abnorm, and perhaps the most exciting one that has been ongoing since about 1994 has been a project that has been utilizing increased databasing to be able to assemble these types of images and correlate them back to what the individual was either looking at, hearing, touching, or in some cases, and this is a stretch, experiencing and thinking, and then trying to match the image through data analysis, which is called a correlational match and fit, to these types of thoughts. They're going one step further, and they're actually doing what's called a spectral analysis of what's showing up here, particularly in the visual cortex, to try to recreate the image with fidelity of what this individual is looking at. They're making some good progress. Now, the limitation, I shall tell you, is that it's pretty easy to do in the visual cortex, because the way this area of the brain is actually oriented and functions. It's a lot more difficult to do when we move out of what we call sensory representation, pure sensory representation, like vision, hearing, perhaps some of the smell areas, and we go into things like, now, what was this person feeling? Although there are plenty of people who will try to do that. We see certain brain areas like these areas lighting up, and what we recognize as these areas that are involved in what we call limbic regions are representative for arousal. Thank God these brain areas lit up when I was thinking about my wife, otherwise I wouldn't be married now. So, yes ma'am?
This is a visual. They're taking a picture of a visual, something.
Yes ma'am.
If you are thinking the same thing without the visual,
this is a great question. The question she asked was the question of representation without identification. So in other words, if in fact I'm looking at this particular thing, please don't let me break it. Good. If I'm looking at this particular thing, is the brain image that is produced when I look at this the same as the brain image when I think about this? And the answer is sometimes. There seems to be a crude representation when the individual thinks about it, but not just in the same place. Because when we think about something, we actually have to conjure up slightly different networks of the brain that then may pull in particular representations, but that's actually more of a memory phenomenon. So what we see is a lot more of these memory-related areas, what are sometimes referred to as the parahippocampal areas, that are then, I hate this word and I'm going to use it anyway, that then light up. Nothing lights up. There really aren't lights in your head, but it seems to get much more active. Yes, sir?
That's a 2D image?
I'm sorry?
That's a 2D image?
Yes, this is a 2D image. This can actually be tessellated into a three-dimensional image that looks very, very nice, and that's called a tessellated image. When we tessellate the image, we can then map this onto a three-dimensional representation. I'm sure you've seen these. They're really lovely. I mean, they make wonderful prints to frame around the house. But the other thing you can do with those is, all kidding aside, is they actually can create a very, very nice hierarchical depiction of the way particular brain regions are activated in time and in sequence. The problem we have with this is that this particular technology doesn't have a lot of temporal fidelity. It doesn't happen real fast. Brains and nerves work a lot faster than this. Why? What is this measuring? This is measuring blood flow and blood use. There's the old adage that I'm sure my neurology and psych friends are familiar with, is that functional MRI is a measure of vein, not brain. And in many ways that may be true. So one of the things we find is that this is time limited. You're sort of getting an after effect. And there is a large literature to suggest that a lot of this stuff may be artifactual, and we have to pump up the volume to be able to pull meaning out of the statistical signal. This, I think, is really, I mean, this is really suitable for framing. I really like these things. And for what I do for a living, this is our bread and butter. This is called diffusion tensor imaging. And when you pair these two things together, what I get is not only a nice image of static areas that are active with regard to blood utilization and use, but also the networks to which they are connected. And I can get a pretty nice temporal signal as well when I then pair this to a physiological measure. What this then does, what diffusion tensor imaging does, is also use a magnetic signal, but it reveals white matter tracts and whole networks rather than just toe bone, foot bone kind of thing. This is sort of like saying, well, we see some activity in Arlington, and there's a little bit going on in Old Town, and Fairfax looks kind of happy too. This comes along and tells you, and by the way, they are connected. So this really shows you sort of the metro system of the brain, and may also provide you with sort of a real-time representation of the way these things might light up when they are co-coupled with other forms of neurotechnology. Alone they don't. Alone this is a static image. But if you then couple these things to serial images over time with neurophysiology, you may be able to determine this in a little bit more what we call viable temporal sequence. But this is nice because it shows that there are real connections that are actually engaged between these different brain areas, and it also shows the relative strength of these connections. So this is nice to be able to sort of give me a roadmap of what's going on in the brain. Where it gets really cool is when I can then couple these images, these snapshot images, to functional activity of the underlying electrochemical networks of the brain. And that's what electroencephalographic measures do. Now, EEG has been around for a fairly long time. These measures tend to be pretty crude, at least they used to be. Now, they've increased quite a bit in their sophistication, primarily through three means. Number one, they use a high number of actual sensors that have a much greater fidelity and specificity. Number two, they're able to translate that program into a high computational program. And number three, they will also couple this to a magnetic field to be able to then create something called magnetoencephalography, which creates a much more accurate representation of the whole network activity of these clumps of cells that may be firing both in unison and out of sync. So what we're able to do is when we pair this with these imaging types, we get a pretty nice picture of structure and structural related activity and function. Although the big limitation here with both quantitative encephalography and magnetoencephalography is that it's pretty much limited to most of the superficial layers of the brain. It really is a measure of what the top layers are doing, although increasingly what we're trying to do is to drill down a little bit, if you will, and I mean that literally, through the use of indwelling brain implants and or some of the use of our nanotechnology sensor capability to be able to then lower the specificity of the signal so that now we're getting a much higher registry in deeper brain levels. That's still a fairly new technology. But one of the things we can do really nicely with that is we can also use a computational program to map this onto a 3D representation, again this is called a tessellated image, of different patterns of EEG activity that we then co-map with either MRI, fMRI, or in some cases a combination of MRI and diffuser tensor imaging. And that gives you this very, very nice structural, functional, what's called analog representation that provides different types of activities with a high level of functional specificity and temporal specificity through EEG, a nice tight time signal that compensates in some ways for the lower signal we get with this stuff, and this stuff. So, take-home message. What do I do with neuroimaging? I compensate for its flaws, and I attempt to delimit its insufficiencies by coupling more than one of these neuroimaging potentials together, or techniques together. And I utilize a program of co-registration that combines more than one of these together with various forms of physiologic imaging, such as this, that can be depicted this way. And I get a pretty nice set of images that can show structure and what structures are engaged in particular activities, behaviors, and cognitions, and patterns of electrophysiological activity. It's pretty nice. On a diagnostic level, as those of you who are clinicians will know, these each and all have their place, both alone and in combination, and we're making some pretty good headway diagnostically. Increasingly, functional MRI is being used as a pre-surgical technique. It's being used as a post-surgical technique to take a look at how the real estate in the brain has been affected by surgery, also by trauma, insult, congenital issues. There's a whole bunch of things we can do with this. But increasingly what's also occurring is that these types of technologies are being used in ways that say, what is the norm for the way particular groups of people will process certain types of cognitions, emotions, and respond behaviorally to a variety of circumstances and situations. Now, you can see that there's a bit of a weakness there. Because what it doesn't do is it doesn't give you predictive value in all cases. So we then move into this beast. This is where we now move into the area of saying not only what's happening right now, but can we look into the proverbial crystal ball of the human genome and your individual genetic complement to determine not only what is, but what will be. And if we couple this with patterns of imaging, could we get a pretty good correlation over a period of time to suggest, yeah, this is what your genes have laid down as a blueprint. And then the blueprint that has been built upon through various sets of environmental experiences, insult, trauma, patterns of behavior, is this pattern of physiological and structural activity that we're then able to correlate an image through some of these neuroimaging techniques that I've just shown you. So what the neurogenetic techniques allow us to do is to understand more about the potential for different brain structures and brain functions to develop, and then correlate what is actually happening in brain imaging over time with the way this potential may have laid down predispositions for certain types of brain function, certain types of cognitions, emotions, and behaviors, and the potential for norms and abnorms, not just physiological. We define what kind of norms and abnorms, as I'll show you in a moment. But what does this do? What do the genomics and genetics actually try to do? Well, I won't bore you by reading the slide, but very briefly what it does is it shows genetic sequences. We all have a particular genetic code. Now there's much of the human genome and our individual genetics that are identical between individuals. But the devils and sort of deities lie in the details of our differences. And one of the things we can find is there are certain genes that are activated and others that are not. And these may then predict, and in some cases, have an influence on defining what brain proteins are then activated for structures, and ultimately may help to then define functions, not only in a sort of static way, but the way the brain and nervous system may respond to a variety of different types of stimuli. So what it does is it attempts to target, remember AAS, assess, access, target, certain genetic sequences that may participate in disease states, conditions, or just simple behavioral and cognitive states. The idea here is what makes you you? Now, before you go, come on, it's pure science fiction. Let me just stop here for a moment. Have any of you ever heard of a company called 23andMe? You have. See, some of you have. We know all the swingers in the room. So have you heard about spit parties? You know what a spit party is? Yeah, see? Okay, what a spit party is that there's a company, one of them is called 23andMe, there are now several more, there's at least 12.5 of these companies, and what you do is you basically spit into a receptacle, send it off for a genetic analysis, and then it comes back with a profile of your particular genetic array, and what it then does is it correlates your genetic array to a data bank, a library of genetic arrays to give you both a description of what you are and have right now based upon the expression of particular genes, and correlates that to a whole set of traits including cognitive, emotional, and behavioral traits. They then give you a little number, a little code. You go to this spit party, and they tell you that your little number, your little code is ideally partnered to individuals with this type of number or this type of code. So what they're doing is they're doing a form of genetic and phenotypic matching. It really is a subtle form of eugenics, but that's okay because what they're really hoping is that you get together and mate, and so this is like the ideal partner. But what they're really doing there is something that I think has sort of sort of factor to it. I can spit, send it off to someone, they'll come back and they can give me a pretty good picture. Now some of it is a self-fulfilling prophecy, it's sort of like what I would call a genetic horoscope, but they can give me a pretty good picture of what at least a blueprint is for my structure and the way I may be predisposed to certain functions. Now, you know, good enough to mate with, good enough to marry, good enough to prosecute? Depending on your view of marriage, they may be the exact same thing. So what this then does is it really helps to predict structure-function relationships from the genome to what we call the proteome to what we then call the neurome and connectome of the way things work in the brain. What is it we're measuring here, folks? What are we trying to do? We're trying to get, pun, our head around this. You'll get this on the way home and you'll laugh. So the idea of this old construct that I learned in school, is it nature or is it nurture? It's both. The idea is that certainly there is a genomic and genetic complement that is expressed, but the differential expression of that occurs through ongoing interactions of that embodied individual with their embeddedness in their environment, internal and external. And if we can assess these different variables at various points in time, we may be able to assess not only what this individual is, but we can then link these to various forms of data to have what's called a correlational prediction of the likelihood of what this individual may do, may be, may become, given interactions with certain types of environments. So what it's allowing us to do is to sort of get a handle on the interiority here, looking at genotypes, phenotypes, structure and function, correlating these things to the body, brain, mind. If you haven't seen this little symbol before, it's called a tilde. It means a complementarity. This is a nice way of me showing you that I'm not of the belief that brains are just sitting out there. I was single for a lot of years, never went on a date with a brain all by itself. Last time I checked, it's in a body. Look around, still holds. Moreover, bodies are embedded in their environments. So we know that there's sort of a complementarity function of the brain and what it does. This word mind, don't ask me to define that. You need to buy me lots of liquor before I'll even go there. And so what we find is that this then affects the state of the body, various sensations, perceptions and cognitions that then lead to behaviors that then come out and affect the way this individual interacts with its environment that then feed back to its interiority and to its exteriority. And if we can get our head around this, and this, and this, we may be able to predict this, or at least that's the idea. That's the idea twice. So, what are we really doing with this? What we're looking to do is we're looking to be in some ways predictive, and in both the medical and the social sense, prophylactic, which is a nice way to say preventive. This idea is not new. The idea of trying to understand the way certain characteristics of genetics, somatic state, and function may predictively lead to individuals who fall outside of some predetermined Gaussian distribution that we, as individuals or a group, call norm. It's quite old. It started way back in the late 1800s with a fellow named Galton, spread all the way up to a fellow named Pearson. And then, of course, we really like to point fingers all around to other countries and go, oh, we know the work of von Verschuer and everything that was happening in the Kaiser Wilhelm Institute that became the Max Planck Institute that happened at the Tiergarten 4 Straße. Whoa, 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 whoa. this is not those nasty Nazis once again. This was a very strong Anglo-European program that tried to identify, can we prevent the harms and ills of society by looking at the structure and function of what individuals are and may be, and pruning that in some way so that these things do not occur. Sort of preventing the thing before it gets too far down the slippery slope, because it's cheaper to do it that way. Well, those are the real and intended pros. And what I mean by real is that this has gotten some definitive use on the medical side. We certainly recognize that genomic medicine, genetic medicine, and the combined use of genomics, genetics, together with pharmacology, various forms of imaging, structural and functional analysis, has been important to helping to define and in some cases predict, and in other cases intervene, genetically determined phenotypic expressions of a variety of different diseases and disorders that we can then jump in early and mitigate. Such things as phenylketonuria disorders, sensitivity to a variety of dietary issues, particular types of congenital hereditary disorders that we can intervene early, and in some cases we may even be able to recognize that there are longer term trajectories. For example, now we're looking at forms of Parkinsonism, various forms of dementia including Alzheimer's dementia, some of the other neurological disorders, saying, well, if we can understand the genetics and the environmental factors, we can intervene early and often, sort of like voting in Chicago, and we may be able to prevent at least the onset or delay the onset to some extent in such a way as to have some viable medical good. But what if the threshold isn't necessarily medical? Or what if the medical threshold spills over and we then say, well, could we do the same thing with regard to the potential for looking at things that may be socially disruptive or disordered? We have to be careful here because there are what we call inherent cons to neurogenetics and neurogenomics. Most notably, that there is a stigmatizing potential. I am not necessarily my genes, and just because I may have a particular genetic complement for something doesn't necessarily mean that I'm going to express it. However, if in fact you're doing serial testing of what we call my proteome and various proteins and metabolites, and you say, well, you're on the road, now just wait a minute. I may be on the road, but I feel just fine. Yeah, but you have, or you are. Yeah, but I feel fine. I don't do X, Y, and Z, but you will. Yeah. So that's number one. Number two is what we call the risk and burden of partial knowledge and we call interventional incapacity. We don't know exactly how all these things work, although we recognize that the idea of one gene creating a disorder is unlikely. More than likely it takes many genes, or what will happen is a single gene may lead to a whole host of potential disorders. But we may not have the capacity to do anything about it, at least not medically. So the idea is if you can't do anything about it, should you even bring it up? And if you can't do anything about it medically, and you still define it as a problem, how are you going to handle that? There's a whole bunch of ways that society has handled that. So if I were to put all this together and sort of draw it into a pretty connect the dots picture for you, what are we trying to do with contemporary neurotechnologies to move towards some ways that are definitive on one hand, predictive on the other, and when I put them both together, definitive and predictive, that leads me to the possibility of being interventionally preventive. And I'm not saying how I'm going to prevent, I'm just saying I'm leading to that threshold. And we're not going to talk about the interventive stuff. You got to bring me back another time and I'll talk to you all about that, and I'll really give you bad dreams. But this is what this looks like. This is what this looks like. This is called the node-edge interaction of neuroscientific and neurotechnological capability for predictive intervention diagnosis. What we do is we take populational genomics and we understand the way these genes express in populations, and then we look at an individual. Where do you fall out in that particular population? Then I take a look at anatomy, and there's a whole bunch of different ways I can look at anatomy. I can look at you and look at what we call crude superficial external anatomy, and I can use a lot of those really pretty pictures to get to things like structural and functional imaging. Not only what are you built like, but how do the parts work, or at least relatively so, given the limitations and delimitations I have in my back pocket and bag of tricks. I then may begin to take a look at chemical biomarkers. This is actually biomarkers, it didn't come up real well. And what I mean by this is I can have a lot more specificity with regard to those chemicals that I can assay, not even necessarily from the brain, but from the blood, from the saliva, from the urine, that have some correlational index of what's going on in the brain. These are called representational biomarkers. The most recent report from Thomson's Reuters listing of biomarkers suggests that there is about 300 to 400 biomarkers that have very, very high validity. And if we use these not alone, but in combination with other forms of assessment, we may be able to get a pretty good picture of what's going on and what could be going on. Then, of course, what we want to say is, this is all well and good, but what are the behaviors? This is the whole idea of sort of the Monty Python thing, is like, bring out your dead, and the guy goes, not dead yet. It's like, well, I haven't done anything yet. No, you may not have done anything really wrong yet, but I'm looking at your behaviors. I'm looking at the way you behave around children, pets, adults, in social circumstances. It's just not quite right. Or we see particular behavioral characteristics, tendencies, and trends that with the right form of very, very highly specific analysis can be sharpened to suggest that these are what we call sentinel behaviors, that if we then get B, C, and D after we identify A, they're going to be yellow flags. If I get F, G, and H, boom, those are red flags. I correlate them with these, and I say, that's going to predict my social dynamics. Now, that's actually where we are right now. Paradigmatically, this is what we are trying to do. Now, that's Minority Report-ish enough, but the good news of that is that we're doing much of this in the area of pathologies of neuroscience, neurology, psychology, and psychiatry into well-identified disorders that apparently have multifactorial issues, contributions, etc., and have been very strongly defined in the past by their behaviors, and sometimes their behaviors alone, and by their social ramifications, one of them being the disorder which is known as psychopathy, formerly referred to as sort of social pathology or antisocial personality disorder. As you may or may not be aware, there's a new diagnostic and statistical manual of the American Psychiatric Association that's due to come out about a year from now. It's supposed to hit the street in early spring 2013. There have been some new categorizations, and much of that relies upon how these are then depicted and a shift in some of the classifications of these disorders. But that notwithstanding, the idea here is increasingly the community to which I belong, which is the neuroscience community, is being asked to do something in the face of such things like Columbine, Oslo, Phoenix, 9/11. There's a public call for this. They look to neuroscience and other forms of science as well, but particularly neuroscience. And can't you figure this out? Can't you figure out what a fill-in-the-blank brain looks like?
What's wrong with you?
Why aren't you getting that grant? Shouldn't you do this? And by the same token, when you say, yeah, we can, or at least get a pretty good idea, you then throw back to them and say, well, what do you want me to do about it? Why don't we just arrest somebody before they do it? You can't do that. No, you can't. At least not in this country, you can't. But what about if they have a high suspicion of... Now, my friends who work in clinical psychology and psychiatry will say, yes, but if in fact there is an increased risk of harm to self and others by virtue of expressed thought, continued ideation, and perhaps representative behavior, yeah, I'm not saying we don't have an out here. I'm not saying there aren't avenues and vectors, little niches and crannies in the gloss of what we call our social agenda that would allow us to perhaps do this. But the question then comes up, what can we really do and what should we do? And one of the things that's important to understand is how we will use these data. When we're looking at individual data, what we're trying to do is to get enough data across various points of the lifespan so that I can look at any particular point that I test you and say, I know what happened before, and I can see the trajectory you're going. That's the idea. Now, before you get the heebie-jeebies and think, oh, this is really creepy, this is the agenda of personalized medicine. There's nothing creepy about that. This is avoid buckshot and let's sharpshoot you so that in fact we avoid the misconception of clinical error and what we call hit and miss empiricism. I know what you're made of, I know your predispositions, I know what you're expressing right now. I can narrow down what I can do like that. because I'm able to utilize these data in such a way as to create a standard to understand what's going on in the, quote, you-ness of you. But not just you, because I can take these individual data across time and lifespan, and I can take a look at cohort data. What about people just like you? Or close enough to you that I can at least clump you together. And then I can compare you to populations of various sizes, including what is now known as the Global Population Data Registry, which you may have heard about, the idea of the Big Brain Data Bank, which is moving to try to get aggregate data of neuroimaging and neurogenomic and neurogenetic data across whole populations to be able to provide these comparative standards. How do we know if these things are tending towards a particular trajectory? Now, we compare them to individuals like you who are close enough that we can say, well, take a look at the same, and she's going in one direction, she went that way before, so you're probably going to go that way too, let's watch. And then we compare them to the whole population. This is what's called a comparative index or a comparative ladder going up. But you know, I can set standards here too. I can utilize what's known as populational indexes and standards to create a normative statistic that then comes down the other way and says, these are my norms, and if you fall outside those norms, then you're abnormal. And the way I do that is like this. This is nothing new. This is the new nomenclature that you will see increasingly in the Diagnostic and Statistical Manual called spectrum effects. Our group, particularly a graduate student who worked with me, her name is Rachel Wertzman, and I spent a lot of time examining the concept, constructs, and phenomenon of the way various things are spectrum presented, going from a genomic representation to genes, to phenotypes, to behaviors. And what we find is that what you see is exactly this. I get a foundation of genetics that through interactions with an environment will produce particular physical characteristics that then will predispose me and allow me to engage in certain behaviors that then feed back in me. And ultimately what I see is I get some manifest effects. But this is an important line. Because although we're striving to define these things biologically, we still have to set the threshold of what we consider to be a cutoff point. When is short too short? I'm serious. We can do this across time. If this were 1890 and this were a pub in 1890, most of the people in the room would be about my size or a little shorter. Now, not so much. What about smart? How smart do I have to be really smart? IQ scores? Biomarkers? How sad do I have to be clinically depressed? How outside some norm of behavior do I have to be so that someone can take a look at my biology and her biology and his biology and go, very similar, it's going to put you on that curve. Let's not forget, these decisional thresholds are both clinical and social, but they are determined by institution and very often by consensus. Now, I'm not saying we shouldn't do this, but I'm saying we need to be careful, number one, about where we set this, and number two, about what types of data we use to determine how to put somebody along this curve. So this is what we're doing right now. This is where we really see the science. What are we doing? We're identifying brain structure and activity that correlated to report cognitive emotional outputs and behaviors, and we're illustrating brain activities that are involved in certain reactions and behaviors. We're making these bidirectional correlations. What are the implications of that? This. We're linking brain states to cognitions, emotions, and behaviors, and we're reaching forward to the potential. And in some cases, we're being asked to scan brains and read minds. Can't you take neuroimages of what these people look like? And if you take enough of them and you put enough people who do these kind of things in a scanner and you get enough of these data bank, well then you can put people in a scanner and you sort of get an image of what brain networks are being activated before they do this. Maybe. Then what? Therapy? Okay. Pull them off the street? For what? How? On what criteria? But there are actually people who are really asking for those criteria to be developed. And not in such a way that is maleficent, but in a way that has beneficent overtones to protecting society at large from individuals who may have biological and psychosocial characteristics and traits that are identifiable biologically and predictive of their psychosocial manifestations. Which then gets us back to this, which is predicting cognitions, emotions, and behaviors from biological data. So what are the issues with this? There are many issues that arise from the technology itself and a pragmatic approach to assessing what the technology can and cannot do, and using that as the groundwork upon which to undergird the prudential question that I'll get to in a moment. These are the issues. What are we really imaging? In 5 or 10 minutes, I went through the pretty pictures and I showed you what these things were doing, and the caveat to each one says, yeah, they're pretty, but this is what they're doing, and we have to be careful when we use this. Obviously, there's also, what are we doing with the objective information? There are certain things that go along with what we call neurocyber issues of data banking. So if we bank enough data, who gets access to these data? For what? And I'll go one step further. This is a very, very useful enterprise, and it's one that I heartily endorse because of its tremendous benefit for clinical medicine. However, my cyber pundits, and I'm not a cyber pundit, tell me if something is stackable, it's hackable. And these data are stackable in data banks and increasingly in clouds. And it doesn't take much to just add one data point to change the entire trajectory, for example, in your genetic code, to then have you represented as being the potential for predisposition of, or particularly even the expression of, a disease state that might have tremendous ramifications for how you're treated socially, economically, vocationally, etc. These then bring in some of the predictive nature of what these techniques and technologies are doing, and the continuous risk of misperception. We have to really make sure that if these are data we're cutting through, that we're utilizing a razor's edge to make these cuts, and not doing so in a broad and cavalier way, because we then fall into what's called naming, framing, claiming, and blaming errors. We name a particular thing as X. We make particular frames of its normality or abnormality. We then make particular claims about what that normality or abnormality predicts and subtends within our psychosocial realities, and then we put the blame. And oh, by the way, what we got to do is take care of that. We need to pull them off the street. We need to make sure they're getting care, or they don't get care. They can't get care. So obviously, these take on not only what we'll call scientific, technological, and pragmatic issues. You then see these opening the door for ethical issues, and of course, the intersection of the practical, technical with the ethical is the groundwork for then the legal issues. Are these issues sufficient to ground legal decisions that deal with things like capacity, capability, culpability, and intervention? And there are certain standards that are extant right now, one that deals with the relative use of these data, which is called Frye standards, and the other that refer to the reliability of utilizing these techniques to make these predictions, and these are called Daubert standards. But these are standards that are decided in judicio. They are decided at the bench based upon the casuistic nature of previous cases that are paradigmatic. And so what happens here is we see these change as a consequence of how these technologies are being used and how often they're being used. Use something often enough, suddenly it becomes the standard. And this is one of the things that's now taking on a tremendous voice in the international neuroethics, legal and social issues literature. There's some good work being done here in this country. There's also an expert center at Friedrich Wilhelm University in Bonn. Really probably one of the best international centers is there. So what we see then is we're facing these very real social tensions.